Hello and welcome everyone to this podcast for cell biology. In this podcast, we're going to talk about cells, and in particular, we're going to spend a lot of time talking about bacterial cells. And when we're talking about bacterial cells, we'll also distinguish the difference between archaea and bacterial cells. And then we'll also spend some time talking about eukarya. Though I'll draw this a couple times throughout the podcast, we can divide our bacteria into a domain up there, and our archaea into this domain, and then our eukarya here. And we're going to begin by talking about features that are common to all cells, and then we'll move into bacteria and archaea and then eukarya. All right, let's go ahead and get started. Next, I want to list features shared by all cells. The first feature is that plasma membranes enclose the cell. In the cell is the cytosol. This is a semi-fluid liquid that fills the inside of the cell. That everything else inside of the cell kind of floats in this cytosol. The third thing common to all cells: chromosomes are present and they carry genes. The last thing here is they all contain ribosomes, which are important in building proteins. Next I want to talk about is a comparison of prokaryotes to eukaryotes. I'm going to write the word prokaryote here, but in a moment I'm going to replace that, because the word prokaryote is not the best word to use. Your book uses it. You may or may not have learned that term in high school, but it's a word we would like to get out of your vocabulary. There was a time when we felt that in this group of prokaryotes there were bacteria and another group called archaea. And we thought these were very similar to each other. They're small. When you look at them under a microscope, they appear very similar. They lack organelles. They're single-celled. Unlike the eukaryotes, which were larger, often multicellular. They contained organelles. So it seemed reasonable at first approximation that this term prokaryote included both bacteria and archaea, which came before eukaryotes. However, we now know that's not true. We now know archaea are actually more similar to eukaryotes than bacteria. So we'd like to stop using this term prokaryote. It's an outdated term, and it implies that these two, bacteria and archaea, are similar, or at least more similar to each other than to eukaryotes. I'm going to draw this in a different way, and this is a way we represent, or can represent, how organisms diverge from each other. We know that at this point here in evolution, there was a common ancestor to all of life. We know that bacteria were the first to form. They're the most primitive. The next thing would have been these archaea. And then finally, eukaryotes. And we can break these down a lot more, and we will later on towards the end of the semester. But for now, what I really want you to focus on is the fact that bacteria and archaea are separate groups, and they should not be lumped under prokaryotes. When we do a genome investigation of archaea and eukaryotes, we recognize and we realize that archaea and eukaryotes are much more similar to each other than either eukaryotes or archaea are to bacteria. So bacteria truly belong by themselves here, as the most primitive, the first cell that formed. And archaea deserves to be its own separate group, as well as eukaryotes. And when I say groups, what I really mean to say are domains. So we have the domain of bacteria, the domain of archaea, and the domain of eukaryotes. And they really are separate groups. I know I'm kind of belaboring this point, but as you move forward, it's best to refer to them in this way and to not refer to the term prokaryote. Alright, now that we've talked about the differences between bacteria, archaea, and eukarya, at least a little bit, let's focus on bacterial cells. We'll begin by drawing a rather large bacterial cell. And really the features of this bacterial cell that I'm going to draw will share many of the same features we would see in archaea. So at this point, they don't look all that much different. The first thing I want to draw is a structure. I'm going to draw it like this, almost as if it's a membrane-bound structure, but it is not. It's just easier to draw it like that. And this is the nucleoid. Nuclei are found in eukaryotes. Bacterial cells don't really contain membrane-bound organelles, but they do have their DNA collected into a single region in their cell that we call a nucleoid. Now within the nucleoid will be a single circular chromosome. Usually, bacterial cells and archaeal cells will contain one chromosome. Let's go ahead and write that down. Here are some features of this. One chromosome, and it's usually circular. This chromosome is the DNA plus proteins that package it together. And this is what codes for all the traits that this bacterial cell may have. Now, the outer side of this bacterial cell, as I've drawn here, is the plasma membrane. Now what I'm going to draw surrounding it, the plasma membrane, is the cell wall. These are these peptidoglycans, the polysaccharides that we talked about earlier in the semester. And I forgot to mention the function of the nuclei. I mentioned it, but I didn't draw it, where chromosomes reside. Now this nucleoid resides in what we call the cytoplasm. In addition to the nucleoid, there are some other features found in the cytoplasm. One of these are ribosomes. And ribosomes are going to be found throughout the cytoplasm. We'll draw those up here. These are important in protein production. Now the next thing I want to show you, I'm going to just draw a couple of them. And these are plasmids. These are also DNA, but they're what we call extra chromosomal, meaning they're not part of the chromosome, they're other pieces of DNA that happen to be in the cytoplasm. The DNA found in these plasmids also contain genes, but these genes are used to protect the bacterium. Sometimes they protect against toxins or antibodies. Okay, now I want to draw what's going to appear as a tail. And this structure is called the flagella, or a singular flagellum. This flagellum at its base sort of acts like a rotor, and it spins the flagellum around, and it provides motility for the cell. Okay, now the last thing I want to draw on this bacterium right now are structures that we call fimbriae. And fimbriae are almost like needle-like projections that extend from the cell wall here. So these are fimbriae. Now I didn't show it, but I guess I could try to indicate it a little bit better. These fimbriae are located all over the surface of the cell wall here in bacteria. Archaea have something similar, but they're not exactly the same thing. These fimbriae sort of act as spikes and help this bacterial cell attach to other structures, either other cells or to surfaces. These fimbriae, in some ways, are like flagellum because they protrude from the cell membrane here, but their purpose is much different. their purpose is to anchor this cell to other tissues or other substrates. And fimbriae, I'm going to write here, source of infection. They really help establish the infective stage of many bacterium. Now in a moment we're going to talk about eukaryotes, and one of the features that we always teach that distinguishes eukaryotic cells, like our cells, human cells, from bacterial or archaeal cells is that bacterial cells do not have organelles. And while that is mostly true, what I want to explain to you are some relatively new findings that suggest that this is not entirely true. So I'm going to write maybe. And that's the process of science. Sometimes we'll teach something for many, many years, and then it turns out that we need to reformulate our ideas. And that's what's happening here. Well, what I mean by that is some bacterial species have what we call primitive organelles. Now, they don't have the organelles that we're used to hearing about in a biology class, like mitochondria, the nucleus, Golgi, or lysosomes, or any of those organelles. So if we were to draw our bacterial cell again, like so, what we are discovering are some small membrane-bound organelles in some bacterial species that will contain enzymes in them within the membrane bound, and they will have very specific tasks. Some of these tasks that these primitive organelles have in some bacterial species include storing calcium. They will hold as crystals the mineral magnetite. And this crystallized form of magnetite will serve as a compass, allowing the bacteria to swim in very specific directions. And in some photosynthetic bacterium, these primitive organelles will contain enzymes that will fix CO2, and we'll talk about that later when we talk about photosynthesis. It'll take CO2 and make complex carbon structures. So they're not the organelles that we're used to thinking about, but they are membrane bound, and they, like all other organelles, they serve a very specific function and help compartmentalize certain reactions. Now, just like the discovery of these primitive organelles in bacteria, we are also discovering that some bacteria have a cytoskeleton. When I was a student, we were taught that bacteria do not contain a cytoskeleton. This is something we only see in eukaryotes. However, research has led us to realize that bacterial cells, at least many bacterial cells in this case, contain a network of cytoskeletal proteins. Now, the cytoskeletal proteins we find in eukaryotes are much more complex. But for instance, cytoskeletal proteins are important in cell division. They help pinch this cell into two separate cells during cell division. Now, we're still learning about these cytoskeletal elements in bacterial cells, but what we're learning is that it's much more complex than we ever thought it was. Still not quite as complex as eukaryotes, but more complex than we at one time thought. I want to spend a little bit more time talking about bacteria, and mainly some of the differences in their morphology. When we think of their morphology, we are going to talk about their differences as it relates to their size, their shape, their motility, and we want to go back and review some of the things about their cell wall, how that is different. And we've already talked about this, but I want to review it again and maybe bring up some other features here. And remember, we're going to talk about the difference between gram-negative and gram-positive cells. But let's go ahead and start with size. I don't want to say a whole lot about size, but I do want you to know that the size of bacterium varies significantly. The smallest bacterium are about 0.15 cubic micrometers. And then the largest are about 200 times 10 to the 6th cubic micrometers. So quite a bit bigger. To help explain how much larger this cell is, the largest bacterium to the smallest bacterium, you could put 1 billion of these. So 1 billion of these bacterium that are 0.15 cubic micrometers could fit in the largest. So 1 billion of these cells would fit inside of this largest bacterium. The next thing I want to do is talk about shape, the general shapes of bacterium. So next I want to talk about the three main shapes of bacterium. The most common of shapes for bacteria are spheres. Another way to describe spheres is to call them cocci, or sometimes we call them round. So they might look something like this, as you would expect something sphere-like or round-like. Alright, the second kind of bacterial shape I want to talk about are the rod-shaped bacteria. These are sometimes referred to as bacilli. They are usually gram-positive bacterium, and these would appear, as you might expect, being rod-shaped, sort of like that first picture I drew of a bacterium. Okay, the last shape we should talk about are the spirals. And these are going to appear to look like something like this, where they actually do spiral, kind of like a corkscrew. You should know these three shapes. Alright, the next thing I want to talk about, as far as how bacteria are different in their morphology, relates to their movement. One way bacteria move, as we've already discussed, is to use a flagellum. And as I described before, you have your bacterial cell here with the flagellum like so. Though we often refer to the flagellum of the bacterium and the flagellum of a sperm as both flagellum, they work very differently. In sperm, the flagellum moves back and forth. But what the flagellum of bacteria do is they act kind of like a motored rotor right here, and they spin this flagellum in either a clockwise manner or a counterclockwise manner, propelling the bacteria in one or another direction. Another way bacteria can move is by gliding. And this allows a bacterium to be on a surface here and to glide, kind of creeping along this surface. Now we understand how a bacterium can attach to a surface, though we don't completely understand the mechanism that allows this bacterium to glide from one location to another location. Okay, next I want to talk about bacterial cell walls. Now we've already talked about these to some extent when we talked about gram-positive and gram-negative bacterium. But let's add a little bit to that. So let's draw the bacterium. And remember that gram-positive bacterium have a very thick polysaccharide around them. And if you remember, that polysaccharide was called a peptidoglycan. Now gram-negative bacteria also have peptidoglycan polysaccharide layering the plasma membrane, but it's not quite as dense, not quite as thick as we see here with the gram-positive. Now in addition, the gram-negative bacterium will have an additional cell wall around it, kind of creating a peptidoglycan sandwich here, where the peptidoglycan, this purple layer here, is sandwiched in between two different plasma membranes, an inner membrane and an outer membrane. Now on a previous podcast we talked about how gram-positive bacteria were typically easier to treat, and gram-negative bacteria were more difficult to treat. Gram-positive bacteria can often be treated with antibiotics like penicillin. Penicillin's role is to disrupt the synthesis of this peptidoglycan layer here. And since it's not protected by this second outer plasma membrane, it's much easier for penicillin to enter the cell to disrupt the synthesis of this peptidoglycan layer. As I just said, this outer membrane here of a gram-negative bacterium makes it more difficult to disrupt the synthesis of this thinner peptidoglycan polysaccharide. In addition, gram bacteria have a channel called porin. Porin allows the transport of things from outside to inside of the cell, but one thing it will do is it will actively pump out penicillin, which makes it harder to treat with penicillin. Now you can treat gram-negative bacteria, but you typically require a different kind of a antibiotic. And with gram-negative bacteria, we typically will treat those with erythromycin. And whereas penicillin goes after the cell wall synthesis, erythromycin go after and poison ribosomes. It makes those ribosomes no longer functional. Okay, so to review, the cell walls of gram-positive bacteria contain a very thick peptidoglycan. Gram-negative contain a very thin peptidoglycan, but an extra membrane that helps protect the gram-negative bacterium against many of our common antibiotics.